Hello, everybody. As Isha said, I'm a New Harvest Research Fellow. Do I have control over this? Yes. I'm a New Harvest Research Fellow and PhD candidate at Tufts University. And today I'd like to talk to you about cellular metabolism as it relates to cultured meat and ways that we can use openly available data and tools to help us understand and potentially direct cultured meat towards the production of a more nutritious product. So in the theme of this session, I'm the user of open data, and I'm going to learn a lot from the other speakers, I think, about the other side. So to start, I'd like to start with this image here, which I'm sure many of us have seen. This is a basic idea of how cultured meat might be produced industrially. So you start with cells of interest, like muscle or fat cells, that are isolated from a livestock species. Those are grown over a series of passages on some substrate or matrix that provides structure, and then you harvest the tissue and you have your meat. In this case, a nice juicy burger. And I'd like to think about this kind of from a new angle for a second, because it's possible to view this process from a more chemical, nutrient-based framework, in which we take molecular inputs in the form of cell culture media. We feed those inputs to cells, which consume and convert them through a range of processes, and then we generate our outputs, our proteins, our fats, our sugars, our aromatic compounds, our flavor compounds. And when we think about cultured meat kind of from this angle, from this input compound to output compound perspective, then the important process really becomes cellular metabolism, or the set of chemical reactions that occur within a cell, converting inputs to outputs and maintaining life within that cell. And in reality, this is a very simple cartoon of what cellular metabolism is, and it's actually a very complex network of reactions. And because of this complexity, researchers have spent a lot of time developing tools to help us understand the various pathways and reactions that make up a cell's metabolism. And one of these tools that I'd like to touch on today is metabolic modeling. So metabolic models are computational models that correlate organism-specific genetic information with molecular mechanisms in order to describe that organism's metabolism. Now, fortunately for the field of metabolomics in general, a lot of these models and the softwares and tools that are used to make and analyze these models are open source. And then fortunately for us in cellular agriculture, there's a long history of trying to understand livestock metabolism for the sake of increasing agricultural efficiency. And so these models exist for species that are relevant to us, like this cattle model here. Now, there are a few applications of these models. They can help us to understand and analyze large data sets, like those about gene expression, metabolic activity, or protein expression. They can help us to diagnose and sort of tease out meaning from experimental results. And in some cases, though this is limited with multicellular organisms, they can serve a predictive role and suggest growth conditions or perturbations that might make cells behave one way or another. And so to sort of talk about what I'd like to focus on today, I'd like to focus on this first method, because there really are a lot of publicly available data sets and these sort of tools out there that we can use as cultured meat researchers. And these data sets weren't necessarily made for the explicit purpose of answering questions that are directly pertinent to cultured meat production, but are really relevant and valuable to us. And I think along with the theme of that session, it's something I'd kind of like to point out and that I think is cool. So to dive right in, I'd like to start by looking at some gene expression data that was produced by agricultural scientists from bovine muscle progenitor cells, satellite cells, that were isolated from a simmental cow, like this one, grown in vitro, and then sequenced at 3 points down the myogenic lineage. So during proliferation, they looked at all the genes that were being expressed. 1 day into differentiation, they looked at all the genes that were being expressed. And then 3 days into differentiation, they looked at all the genes that were being expressed. And what we can do is we can take this data, we can map it against this bovine genome scale metabolic model, and we can see this sort of heat map of expression and of expression levels throughout all these different metabolic processes. And we can then look kind of through this data that was produced by other people for trends that are interesting to us. So for example, if we look down into this corner, into the region of lipid metabolism within this model, so fat metabolism, we can see that the genes involved in the synthesis of saturated fatty acids seem to be expressed at different levels throughout the myogenic process. And indeed, when we kind of dive into that data, we see that fatty acid synthase, which is involved and responsible for the synthesis of saturated fatty acids, is expressed significantly more higher on day 3 of differentiation than it is on day 1 of differentiation or during proliferation. And now for the traditional agricultural scientists who produce this data, this trend might be interesting, but it might not necessarily lead to design considerations that they want to take into account when they're thinking about producing a product. For us, though, the trend can take on new meaning. And specifically, it can take on new meaning when we think about the nutrition of saturated fatty acids. So high dietary intake of saturated fatty acids, which is shown by the bottom line here, has been associated with increased cardiovascular risk and increased mortality in humans, which we can see by a loss of cumulative survival over time. On the flip side, high dietary intake of unsaturated and particularly polyunsaturated fatty acids like the ever-famous omega-3 fatty acid, that's been associated to reduced triglyceride levels in humans and therefore improved cardiovascular health and reduced mortality. And so taking these two facts into mind and thinking about the fact that when we're talking about cultured meat, we can control how we grow our cells, how and when we choose to differentiate cells and start them down that myogenic lineage, and how and when we choose to harvest our tissue, we can kind of develop a nutritional goal for our product. And we can say that we want to decrease the levels of saturated fatty acids in the meat that we're producing, and we want to increase the levels of polyunsaturated and omega-3 fatty acids in the meat that we're making. And because we have control over how and when we grow and harvest this tissue, the data that we were looking at points towards a possible solution. We can potentially harvest cells earlier in the myogenic lineage in order to get cells that have gone less far down the differentiation process and presumably have produced fewer saturated fatty acids as they kind of move down that myogenic lineage. So I think that this is a promising option that is suggested by that data. It definitely has some drawbacks. So for one, it wouldn't lead to increased amounts of omega-3s in our meat. And for another, growing or harvesting early on in the myogenic lineage might have effects on flavor or texture or other nutritional compounds, as we heard from the talk earlier today. So we can instead think about this from an input-output compound perspective. And we can think about adding omega-3s to our media. They'd be taken up by cells, healthier meat, healthier people. We've achieved our goal. In this case, there are potentially added costs of introducing another media component that could drive up the price of the final product. So that's sort of a downside there. And then finally, we can think about internally converting saturated fatty acids into unsaturated omega-3 fatty acids within our cells. Now, the enzymes that do this for omega-3s don't naturally occur in mammalian cells. So the question becomes one of genetically engineering our cells using enzymes from an organism that can produce these omega-3s. Unfortunately for us, researchers have previously shown that fatty acid desaturase genes from a C. elegans nematode expressed in mammalian cells make it so that those mammalian cells are able to do just that. And they can convert saturated fatty acids into unsaturated omega-3 fatty acids. And just sort of as proof of this, the red charts show increased omega-3 content in cells expressing this gene. And to the left, we see a decreased level in more highly saturated fatty acids in these cells. And so building off this idea, what we've done is we've taken these same fatty acid desaturase genes, we've inserted them into a vector with a mammalian promoter and the gene for green fluorescent protein, as well as a little linker that I'll get to in a second. We've inserted those cells into mouse muscle cells, or we've inserted those vectors into mouse muscle cells, and we've grown the cells and see what happens. And so in theory, what happens here is you get simultaneous expression of those fatty acid desaturase genes along with green fluorescent protein. You get simultaneous translation of those two proteins, and then after translation, that little linker in there breaks, and so you're left with two distinct proteins. And because you're getting simultaneous expression, and because the genes are ordered so that green fluorescence is dependent on the expression of fatty acid desaturase, we can have a fairly good indicator that if our cells are green, they might also, and I'll just leave it at saying we have a good indicator, that they might be producing that fatty acid desaturase as well. And so indeed, when we look at these cells, we see that they're fluorescing well, they're producing this green fluorescent protein. And importantly, we see that they're able to continue down the myogenic lineage and differentiate into myotubes, which means that with a method like this, we wouldn't necessarily have to take into account the same flavor, texture, sort of potential confounding factors that we would with, say, an early harvesting protocol to try and target these fatty acids. Now, there's definitely further work to be done on this. We need to make sure that the enzyme's actually there and actually active, and then we need to move this system into bovine muscle and fat cells. But I think that this is a good sort of example of a way that we can tailor the lipid profile of our muscle and make a more nutritious product. So to take a step back, I'd like to look back at our whole metabolism and think about another metabolite that we might want to target to improve the nutrition of a cultured meat product. And specifically, I want to look at this metabolite here, carnitine. So carnitine is an important component in all cells, but high dietary carnitine has been linked to numerous pathologies, including arterial thickening, aortic lesions, and type 2 diabetes. And unfortunately, for beef eaters, red meat is notoriously high in carnitine, higher than any other form of meat, much higher than any vegetables, fruits, grains, dairy products, what have you. And so, as the sort of, I guess, self-acclaimed cultured meat nutritionists that we all now are, we can develop a new goal. And we can say that we want to decrease the levels of carnitine in the meat that we're making. And again, we can look at this data that was produced not to answer this question, but made available to us and can prove useful to us. And we can see that as with saturated fatty acids, synthesis of carnitine and the expression of the genes involved in carnitine synthesis increases the further we move down the myogenic lineage. So again, we might want to think about harvesting early on in the process of differentiation, or maybe even during proliferation, to sort of target this metabolite. Obviously, again, we need to sort of consider those same potential negative implications. But I think it's another promising option provided by this data set. And then we can also think about this from the input-output perspective. And we can think about tailoring our media and the amino acid precursors to carnitine that are in our media and tailoring levels of those to affect the subsequent carnitine production. And when we do this in those same muscle cells, we see pretty clear results. We see that when we double the levels of amino acid precursors in our media, we get a 5-fold increase in carnitine, in cellular carnitine, compared to controls where we double levels of another amino acid. Interestingly, this trend doesn't continue when we quadruple the levels of our amino acid precursor. This could be due to cell death, as we see that increasing levels 100-fold results, it could be due to cell stress, because we see that when we lift it up 100-fold, we get cell death. Or it could be some other metabolic phenomenon going on that a model might help sort of diagnose, as I mentioned earlier, possibly. But I think that this is sort of I wanted to mention this kind of as a clear picture of a way that we can tailor media components, not just to think about improving the growth of these cells, but to target specific metabolites that we do or don't want in our muscle from a nutritional standpoint. And so to conclude, I hope that I've been able to emphasize that cellular metabolism is extremely important when we're talking about a product that's so defined by its composition, its flavor compounds, its nutritional compounds, its aromatic compounds, and how metabolic models and these tools that have been developed by other researchers can help us sort of visualize and understand all the different reactions involved in this complex process. Second, sort of on the theme of this session, I really want to highlight that there are a lot of openly available data and tools like RNA sequencing data or metabolic models that are out there that we can use in cellular agriculture in order to save on the cost of producing RNA sequencing data ourselves, or just to kind of have a larger wealth of potential resources than we might have in this very nascent field. And then finally, I just want to reiterate that it's possible to tailor the nutritional profile of this tissue and affect the nutritional profile of this tissue using a range of options, such as genetic engineering or leveraging changes in metabolic development or in metabolic activity throughout development, or tailoring our media. And I think that this is a really promising option for adding value to the product and making something more nutritious than conventionally produced meat. And so with that, I'd like to thank a few people in particular. Thank you to Dr. David Kaplan, my advisor, and to everybody at Tufts University who's helped in my first year. Thank you to the New Harvest team and to all the other fellows for all your questions and advice. Thank you to Dr. Jonathan Dreyfus of the Joslin Diabetes Center, who has been extremely responsive to questions that I've had about metabolism and metabolic modeling. And then thank you, all of you, for your attention.